So when RNA polymerase II first transcribes mRNA, it doesn't look like this nice colorful one here. It's first called heterogeneous nuclear RNA, or hnRNA, and needs to go through a few more processing steps in the nucleus before it's officially an mRNA. First, a 7-methylguanosine cap is added to the 5' end, which is literally just a guanosine molecule with an extra methyl group on it, but connected backwards. So its 5' end is connected to the 5' end of the mRNA by 3 phosphates, as shown here. Next, the sequence AAU-AAA at the end of the 3' UTR is used as a signal to add on about 200 more A's to the end, which is called polyadenylation. Both the cap and the polyadenylation regulate nuclear export, prevent degradation by exonucleases, and promote translation. Third, introns have to be spliced out, which I'll go through in more depth in the next slide. But before we go on, can you think of any examples of genes or types of genes that do not receive 5' caps? Since the 5' cap is only added in the nucleus, genes in the mitochondrial genome would never be capped. Okay, let's talk about splicing. This is how introns are removed from hnRNA. We have this nice diagram in the book, so I'll use that. In this diagram, the intron is the DNA between exon 1 and exon 2, and this includes a few key nucleotides. First, the transcript combines with the small nuclear ribonucleoproteins, sometimes called SNRPs, to form a structure called the spliceosome. The SNRPs recognize specific sequences in the transcript and cause the DNA at the beginning of the intron to detach from that exon as this phosphate binds to a specific adenine within the intron, forming a loop called the lariat. Here you can see what it looks like once the loop is formed. Exon 1 is then joined to exon 2, and the lariat is no longer attached to anything, so it floats away and is degraded. This lariat contains the intron, so after doing this to all the introns, you end up with only exons. The concepts here are more important than the details for your exam, but one detail that could be tested is that there's a certain disease in which patients make antibodies to snRNP proteins. Any idea what this is? This would be lupus. We've already covered a lot about introns and exons, but one last thing I want to cover here is the concept of alternative splicing. So you've got all these exons, but you don't need to use all of them when you splice them together. Exons can be combined in different combinations to create different versions of the protein, and exons often correspond to specific protein domains. For example, maybe there's a protein that has a DNA binding domain, a catalytic domain, and 2 regulatory domains, so it can be repressed by other proteins. If for some reason a cell only wanted one of these regulatory domains to be present in certain situations, it can do that by keeping or removing that exon in the mRNA. This saves a lot of space in the genome compared to having 2 entire separate genes, which is especially useful for viruses since they have a very limited space for their genome. Alternative splicing is another area in which mutations can cause disease though, with a key example being beta thalassemia. In beta thalassemia, a gene that codes for the beta-globin protein in hemoglobin has a mutation in part of the gene that normally allows the spliceosome to recognize the intron-exon boundary. The mutation prevents proper splicing, so you end up with an incorrectly spliced mRNA which codes for semi-functional or dysfunctional protein. Alright, for our next flash quiz, let's revisit mRNA modifications. So is the 5' cap added to the mRNA before or after it leaves the nucleus? So this would be before it leaves the nucleus, because remember, the 5' cap, just like the poly A tail, is necessary for export from the nucleus. Now let's move from mRNA to tRNA. The tRNAs are about 75 to 90 nucleotides long and form this cloverleaf structure, which you can see here. The 3' end is called the amino acyl end, since it's where the amino acid is bound, and it ends in the sequence CCA. You can think of this as standing for can carry amino acids, since this is where the amino acids attach. The opposite end contains the anticodon, which is complementary to a codon in mRNA. It's this matchup that allows each codon to correspond to a specific amino acid. Before it can do its part in translation, a tRNA must first be charged by adding its corresponding amino acid to it. This is an ATP-dependent process, catalyzed by amino acyl tRNA synthase, and there's a different enzyme for each tRNA. So here we have a tRNA that corresponds to methionine, so this might end up with a start codon, and the amino acyl tRNA synthase for methionine will first check if this is indeed the methionine tRNA, and if it is, it'll attach a methionine amino acid to the 3' end. This step requires 1 ATP. Once the tRNA is charged with its amino acid, initiation factor 2 is needed to get it into the ribosome so it can find its codon. This is a vital process, so we can use antibiotics such as tetracyclines that target the 30S subunit of the bacterial ribosome and prevent attachment of the amino-acyl tRNA, and this will actually kill bacteria. I talked about tRNA wobble a few minutes ago, but I'll briefly go through it again. Basically, the idea here is that there are many amino acids for which multiple codons can all be used to get that same amino acid. For example, proline is coded for by all of these codons, which differ only in their third base. So if there's a mutation in that third base, you'll still end up with the same amino acid in the protein. Therefore, this third base doesn't matter as much as the other two. Now, this isn't always the case. There are some amino acids that are only encoded by one codon, but by and large, the ones that are encoded by multiple codons will only vary in the third position. Okay, on to the last major process in this section, protein synthesis. Protein synthesis occurs in 3 steps: initiation, elongation, and termination. Initiation involves the assembly of the mRNA, the 40S ribosomal subunit, and the initiator tRNA, which will almost always be the tRNA loaded with methionine. There are 3 different sites in the ribosome. The A site is where all amino-acyl tRNAs, or tRNAs that have amino acids on them, will first come. The only exception is for this first one for the start codon, which starts at the P site. Proteins called initiation factors are needed to put these 3 things together, and they use GTP as an energy source. Once the mRNA, tRNA, and 40S ribosomal subunit are assembled, the 60S ribosomal subunit joins the party, and the initiation factors leave. Now we can start the next phase called elongation. The P site is where the growing peptide will be, and the A site is where our next tRNA will come in, which has an anticodon that's complementary to the codon after the start codon on the mRNA. So which end of this mRNA is the 5' and which is the 3'? In this case, the left is 5' because that's the direction of the beginning of translation, and the right is the 3' end. After this tRNA has arrived, the ribosomal RNAs will catalyze a peptide bond formation between these 2 amino acids, and the amino acid that was in the P site will be set free from its tRNA. The last part of elongation is called translocation, and this is where the ribosome moves 3 nucleotides toward the 3' end of the mRNA, so the tRNA that's attached to the polypeptide ends up back at the P site. The A site is now temporarily empty, and the disconnected tRNA is in the E site, which you can think of as standing for empty or exit, since the tRNA is now free to go. This cycle will repeat while the ribosome goes all the way through the regular codons, but I won't show all that here in the interest of time, so let's pretend that this protein only has 2 amino acids. Eventually, the ribosome will reach the stop codon. This brings us to the last step in protein synthesis, which is termination. And as I mentioned earlier, the stop codon, which in this case might be UAA, is recognized by release factors, and the finished protein is released from the ribosome. Okay, so once again, what do you need to know about any of this as a clinician? Well, the most relevant part for your boards is drugs that target translation. The bacterial ribosome is different from the eukaryotic ribosome and has 30S and 50S subunits instead of 40 and 60. Since it's different, we can use drugs that target it specifically and have little or no effect on our ribosomes. For example, aminoglycosides inhibit initiation by binding the 30S subunit, chloramphenicol inhibits the peptidyl transferase activity of the 50S subunit, and macrolides prevent the release of a tRNA that has already donated an amino acid, all of which prevent protein translation and therefore kill the cell. These are discussed in more detail in the microbiology chapter. Alright, here's the last flash quiz for this section. The first tRNA used in protein synthesis, which carries methionine, will enter the ribosome and go to the A, P, or E site. The first tRNA will go to the P site, which is an exception, since all subsequent tRNAs will start in the A site, and only move to the P site after their amino acid has been added to the growing polypeptide. So once a protein is produced, it's usually modified before it's in its final form. One way this is done is by chopping some amino acids off the end, which is called trimming. This is common especially in dangerous proteins such as digestive enzymes, where you don't want them to be active until their target is around, since they could damage parts of the cell or surrounding tissue. The enzymes themselves are produced in an inactive form called a zymogen, and a big piece has to be cut off by another enzyme for them to be activated. There are a few smaller covalent modifications that can be added to proteins too. Phosphate groups are added or removed to turn a lot of enzymes on or off, like a light switch. Glycosylation is often used to change what a protein can and can't bind to, and hydroxylation is essential to form strong bonds between collagen molecules. As I mentioned at the beginning of the chapter, histones are methylated and acetylated to regulate expression of nearby genes. Do you remember what effect acetylation usually has on gene expression? It generally increases transcription, whereas methylation of histones usually represses transcription. Lastly, when a protein is damaged or no longer needed, it can be modified by adding a small protein called ubiquitin to it, which tags it for degradation by the proteasome, which we'll talk about more in the next lecture. This final slide in the molecular section of biochem is about chaperone proteins. We're a little bit out of order here since chaperone proteins do their job before post-translational modifications. Alright, but what do chaperone proteins do? Well, one thing that we don't really talk about in the book is protein folding, which is a pretty important topic, although honestly not very likely to show up on your step 1. The amino acids in proteins fall into a few different categories, and we'll get into this more in the metabolism section, but they can be negative, positive, or uncharged, and they can also be polar or nonpolar. The polar ones like to hang out together, and the positive ones like to stick to the negative ones, and vice versa. So we have to get from this sequence of amino acids into a complicated three-dimensional shape, like our ubiquitin protein here, which by the way is not a chaperone protein. So how might these fold? Well, glutamate is negatively charged, and lysine over here is positively charged, so maybe they'll try to stick together. But wait, the arginine at this end is also positively charged. So how does the glutamate know which one it should be attracted to? It's like a molecular love triangle. So ultimately, the protein will be happiest in a certain shape, but sometimes needs a little bit of help to get there. So here's a question for you. If you have a protein floating around in the cytoplasm of a cell, do you think the polar amino acids like to be on the outside of the protein or buried in its core? Well, the polar ones like to be around water, so they'll be on the outside. However, if there's a protein with mostly nonpolar amino acids on the outside, where do you think you might find it? This would probably be embedded in the cell membrane, since nonpolar amino acids are happy around lipids. So here's an example, a sodium-potassium pump. Chaperone proteins are there to direct folding of other proteins so that they end up in the right structure. Depending on the protein, they can do this in a few different ways, but one way is to form a large, multi-protein chaperone structure, kind of like a barrel, that newly synthesized proteins can be imported into, and once inside, they're able to fold much more easily because the polar amino acids in the inner wall of this barrel help to push all the non-polar amino acids in our new protein towards the inside. Alright, so why are chaperone proteins important? Well, first of all, improperly folded proteins can cause disease. Things like protein aggregation that you see in Alzheimer's disease or amyloid deposits that you learn about in the pathology chapter are caused by misfolded proteins. The last thing I want to touch on are a type of chaperone proteins called heat shock proteins. In a hotter environment, particularly in single-celled organisms or cold-blooded animals, but also in humans to some extent, proteins are less likely to fold properly. So heat shock proteins are created in response to heat shock, like if you jump into a hot tub, to prevent misfolding of proteins. The example we give in the book is heat shock protein 60, so named because of its 60 kilodalton size. And this protein is used in the structure shown here. Do you recognize this? Right, that's a mitochondria. So remember, most proteins in the mitochondria are made outside of the mitochondria and have to be imported into it. And once they get there, heat shock protein 60 helps them fold into the right shape. Alright, that's it for the molecular section. When you're ready, you can move on to cellular.